Thank you so much for the introduction. We are going to talk about the work that we develop with pluripotent stem cells as potential tools to evaluate the development of drugs, specifically the safety. As Elias mentioned, I am in CEDAR, Center for Drug Evaluation and Research. So I'm going to start talking about technology for assessing the risk of drugs. And I'm going to travel in time and go to the 1930s. In the 1930s, this was the state-of-the-art technology for aviation. This was the state-of-the-art technology for commercial automobiles. And this was the state-of-the-art for preclinical tests of drug safety. Now let's come to 2017. State-of-the-art technology in aviation are jet planes. Now cars are much more efficient, safe, and nothing much has changed in terms of technology for evaluating preclinical safety of drugs. So this is where the division where I do research at the FDA comes up. So in the division of applied regulatory science, our vision is to move new science into the drug regulatory process to change paradigms that haven't been changed in a very long time. And we do this by performing mission-critical applied research to develop and evaluate tools, standards, and approaches to assess the safety, efficacy, quality, and performance of drugs. But also, we perform expert regulatory review consultations for mechanistic safety evaluation for intermediate regulatory needs. And this can be a critical review of existing knowledge on certain types of compounds or drugs, computational analysis, in vitro, in vivo laboratory testing, or translational analysis from preclinical studies. So in our division, we do translational research. We collaborate and do interdisciplinary team approaches. And our goal is to implement all of this in new regulatory review methods and programs. And we collaborate with many offices in CEDAR, with different centers in the FDA, with other federal agencies, as well as with industry and academia. Overall, our goal is to basically bridge the science and discovery that happens at the academic level and bring it to the regulatory process to improve the evaluation of drugs, specifically safety. And specifically, in our labs, we predict the clinical toxicity of drugs with pluripotent stem cells. Now, pluripotent stem cells. Many of you may not know what these are, so these couple of slides are dedicated to explaining what they are and how can they be used for evaluating drug safety. So you see on the slide an image of a cell, a normal cell, a regular cell. It has a nucleus, it has the cytoplasmic membrane that surrounds the cell, and then the intercellular organelles. And pluripotent, pluri means several, and potent means being able. So pluripotent stem cells, they are able to generate any type of cells in any tissue that exists in the human body. And this happens through different sets of pathways that can be regulated in the lab. Now, today from my portion of the talk, the key takeaways are that these pluripotent stem cells have high potential for predicting clinical drug toxicity. However, engineered microfabricated systems can address the main problems of using these cells. And the problems of using these cells is the fact that when differentiated, these cells are still physiologically immature, and there's a lot of variability between cells, between cell lines. And the main focus will be on structural maturity and genetic variability. Now, there are two types of pluripotent stem cells. There are the embryonic pluripotent stem cells and the induced pluripotent stem cells. So the embryonic pluripotent stem cells are isolated from a blastocyst embryo, more specifically from the inner cell mass where we all originate from. The induced pluripotent stem cells are reprogrammed from somatic cells. These can be blood cells, can be skin cells, can be fat cells, can be any kind of somatic cells. And the interesting thing about this is that they're isolated from a consenting adult, not from an embryo. And they can model patient-specific features, and therefore they have high potential for precision medicine. This is precisely the reasons why we're focusing a lot on these specific pluripotent stem cells, and from now on, everything that we'll talk about in terms of pluripotent stem cells regard induced pluripotent stem cells and not embryonic pluripotent stem cells. So I don't expect you to even be able to read what's in this figure. This figure is just a bunch of arrows, and in the center you have induced pluripotent stem cells that can, through these arrows that represent different pathways and different experimental conditions, they can originate any cell in the body. They can originate liver cells, and we can do that in the lab. They can originate bone cells, and we can also do that in the lab, as well as, neural cells. So in a regulatory setting at the FDA, in CEDAR, we care about modeling tissues and working with cells that normally represent the common targets of adverse effects of drugs, of the adverse toxic effects of drugs. So I have a poll question. What are those? What are the specific, the typical tissues that are normally affected by adverse effects? Is it brain, liver, Is it brain, liver, heart, bone, kidney, pancreas, lung, blood? These polls can only happen online. I apologize. But you feel free to say, anything if you feel like, well, this is going on. So most people are saying liver, kidney, and blood also a bit. No one is saying bone. And brain also, some people are saying brain. Okay, I think this is good. So one, brain one, right? No, liver one, yes. Yeah, so these were the different options in the poll. In fact, heart and liver are the most affected tissues, organs in the body from drug adverse effects. This talk, we will focus in the heart, both of the talks that will be given here. And because, heart cells, we'll focus on heart cells. So pluripotent stem cells, as are shown on the left image, can be differentiated into cardiomyocytes, the cardiac cells, the muscle cells. This involves, in the lab, changes in culture media, different small molecules, and purification steps to make sure that we have a pure population of what we want. And this is kind of a graphic of, diagram of how the timeline of what's going on. It takes 30 days, and that's kind of the key message, is that this takes some time to happen. But the ultimate goal is, once we have these cells that they're isolated from a human, reprogrammed into pluripotent stem cells, and then differentiated 30 days after 30 days, is to have these cardiomyocytes and use them for testing safety of drugs. And specifically, we want to do assay for drug toxicity. So how do we assay function of a cardiomyocyte, of a heart muscle cell in a dish? So on the right side, you have a video of a mouse cardiomyocyte, which is very similar to a human cardiomyocyte. This is not differentiated from a pluripotent stem cell. This is isolated from a heart. So it's the real deal. And as you can see, it has a particular shape of the state of Tennessee. And also it has these fibers that are aligned along the main axis, right? So these are kind of typical properties of cardiomyocytes. But also they have a physiological function. They beat, as you can see. This beating happens because of an electrophysiological activity. They have calcium flow, and of course they need energy. They have also typical metabolic properties. And none of this would happen without a well-regulated genetic expression. So one thing about differentiated cardiomyocytes, not these isolated from the heart, is that they should be mature. What does that mean, to be mature? So this is another image. I apologize, the contrast is not very good here, of an adult cardiomyocyte isolated from a heart. In green, you have Z lines, and in blue, you have the nucleus. And again, the cell has the rectangular shape of the state of Tennessee. And these Z lines, basically in series, they have all these proteins that are the contractile machinery. They have the machinery that allows the cell to beat. And in series, they form aligned fibers. And these fibers are aligned in mature cardiomyocytes, and the Z lines are well registered versus low registered. Now, if we look at a cardiomyocyte differentiated from a pluripotent stem cell, you see something completely different. The shape is lost. They have a random shape. But also the organization of these fibers and Z lines is also lost. In fact, they resemble much more fetal cardiomyocytes or neonatal cardiomyocytes. This means that they have non-aligned fibers, lower registry of Z lines, and therefore they are immature, clearly. So something that we do a lot in our labs is to use microfabricated devices to mature these cardiomyocytes differentiated from stem cells. Now, microfabricated devices, it's another term that's not known to the common public, but basically what we want to do with this is use different techniques, can be polymer chemistry, material sciences, surface chemistry, soft lithography, etching, etc., micromolding. And what we want to do is to use combinations of techniques to modify the dishes or the plates where the cells are cultured to make them more mature. And we do this by first understanding the physiological conditions of the heart or of the tissue that we want to model. And this can be the three-dimensionality, the mechanical stress, cytokines, presence of other drugs, nutrients, etc. And then we want to, with microfabrication approaches, replicate physiological settings in a dish to enhance maturity. And one central, I mentioned shape as being one of the central keys for cardiomyocyte maturity. There are many microfabricated approaches to improve the shape of these stem cell differentiated cardiomyocytes. You can do that by microcontact printing, by substrate topography. We can make three-dimensional microtissues that are aligned. We can stretch cells and make them aligned. And we can change the elasticity of the substrate that induces cells also to align. All of these can align cells. And aligned cells have a more physiological structural organization and have a more mature function. Now, we also hypothesize that they're suitable for toxicity assays. Now, something that I'm going to talk about now is what happens when we combine microcontact printing with substrate rigidity. So on the left side, you have cells with a random shape and random organization of those structures, those Z lines. And what happens, and I really apologize for the poor quality of the images, what happens when we put them on micropatterns with a rectangular shape and also on a material that has the same rigidity of the heart is that we improve the maturity of their shape and also improve the alignment of the fibers and the Z lines. So what do we do with these microengineered cells that are more mature? So this is how we envision the use of microengineered cells and microengineered tissues. We differentiate cells, which in this case takes 30 days. We add them to engineered systems that make them more mature. Then we apply a measurement technique, we get data, and then we quantify biomarkers from this data. And we basically use this to evaluate drug safety with different drugs. Nowadays, people bypass this. However, by implementing engineered systems, we improve the physiological relevance of measurements, and we mature cells by delivering different types of stimuli, chemical, electrical, biological. Now, before I wrap up my part of the talk, I want to talk to you guys about variability, specifically about genetic variability. So on this table, you see on the left side different types of variability that can happen with pluripotent stem cells. You have functional variability. It's basically, you know, if you're looking at contractility, it's the way they contract. If you're looking at calcium, it's the way calcium flows. Production of messenger RNA, epigenetic variability, and genetic variability. And on the right side, we have different types of variability that can exist. So within a specific type of embryonic stem cell, within a specific type of induced pluripotent stem cell, and between embryonic stem cell and induced pluripotent stem cell, and just heterogeneity within the line. And the plus and minus means high variability plus, minus low variability, and the question mark is unclear. So as you can see, for the different types of variabilities and the different ways you can analyze variability, you see a lot of pluses, which means that there's a lot of variability, and, you know, also a few question marks. There's actually one minus on this table. This means that, you know, this is a big problem if we want to be confident on the data that we acquire from these cells, mainly for evaluating drug safety. So, you know, main takeaway of this table is that gene expression is highly variable, and as I mentioned before, function follows gene expression, so that one also will be variable. I want to talk about a specific paper that came out earlier this year that showed that of all this variability that has been known to occur in pluripotent stem cells, only 50% is regulated by variability between individuals. In other words, if I isolate the pluripotent stem cells from me or from one of you, only 50% of the variability between the pluripotent stem cells from different individuals is because of the genetic differences between the individuals. Everything else is regulated by other mechanisms. And they looked more into these mechanisms. They identified a set of genes that regulates variance, another set of genes that regulates magnitude of variance, and they also saw that differentiation is affected by the variability of pluripotent stem cells. So I want you just to focus on the big pie chart. And as you can see in the green, you see the amount of unknown factors for causing variability. And this is why this field still requires a lot of research. The biggest problem is that, in our case, is that populations of differentiated cardiomyocytes are highly variable. And what do I mean with this? So in this figure here, as you can see, again, I apologize for the poor quality, you see in gray or white, it's the DNA, it's the nucleus. So each cell is represented by a nucleus. And in green, you have the Z lines. So what you see in the figure is some cells have well-developed Z lines, some cells have poorly developed Z lines, and some cells, you see a lot of nuclei, they don't even have a green. They do not co-localize with the green, so they don't even have Z lines. This is the type of variability that people that work with these cells constantly see. So how can microfabrication also help study variability? What we can do with microfabrication, we can actually look at single cells. We can pattern cells individually in an array and look at each cell at a time with image-based approaches. And in this video that you see on the left side, you have a single beating cell, but we can actually look at all the cells in an array in a dish. We can look at thousands of cells. And what we can do, we can look at the cells that, can label the Z lines in these cells. We can look at the cells that are mature. We can look at the cells that are defective and, analyze different cultures, different lines, different differentiation protocols. But then we can take the way of the future and look at these single cells to do single cell studies. Look at function, contractility, electrophysiology, calcium, and relate this for the same cell. We can do omics studies, genomics, proteomics, metabolomics, etc., all the omics. And the goal is to learn how to select for a mature cell. What are the properties of a mature cell? And to test the hypothesis that, a set of cells, of mature cells, can give us robust and consistent results when evaluating drug effects, while immature cells or cells that are more variable cannot. So I hope I was able to convince you that induced pluripotent stem cells have high potential for modeling heart function in vitro, and that engineered microfabricated systems can solve the main limitations of these cells, which are lack of structural maturity and high genetic variability. I'd like to open the floor now to Ksenia Blinova, where she's going to talk about her recent work in studying proarrhythmic phenotypes in cells. Thank you so much.
Thank you, Alex. Good morning, everyone. I am Ksenia Blinova. I work for FDA's Center for Devices and Radiological Health, where I lead an induced pluripotent stem cell and electrophysiology core facility. In my talk, I will build upon Dr. Ribeiro's introduction of this exciting new technology to describe how regulatory research conducted at FDA can have an impact on medical product development and approval. So Center for Devices, or CDRH, is a very large center that oversees numerous medical devices, and in addition to several offices that deal with pre-market submissions and post-market monitoring, we have a very substantial office of science and engineering laboratories, or OSEL, where regulatory research is done. Our mission is to make sure that the center is ready to make science-based decisions, especially when it comes to emerging and innovative technologies. To give you a scale, Division of Biomedical Physics, that is one of 4 research divisions at OSEL, has about 100 scientists, mostly PhDs, MDs, and engineers. So the range of research areas that are covered by our division is very broad. We have a large optical physics group that works with ophthalmology devices, optical imaging, and laser safety. Neuroscience group has its 3D motion capture equipment used to assess gait and hip implant patients. This is in virtual environment, including EEG to study prosthetic function. The images on the bottom are for cortical electrostimulation that can be used in computer-brain interfaces, for example. And here they assess the electrode-tissue interaction for the implantable electrodes. This is an anatomically correct whole-body computer models for electromagnetic and thermal stimulation developed at FDA in collaboration with academia and used by industry in many successful pre-market applications. It is used, for example, to model the heating from deep brain stimulation leads during MRI scan. The division also hosts a 3D printing facility that can be used to tailor the shape of a device or implant to a specific patient anatomy. And finally, my group that focuses on cardiac models. Today I will be talking about how stem cell-derived cardiomyocyte and high-throughput electrophysiological devices can be used to test the potential for new drugs to induce cardiac arrhythmias. So I will start by reviewing the current regulatory approach to the prevention of drug-induced arrhythmias. Then I will explain how and why the current approach can be improved with a new comprehensive in vitro Proarrhythmia assay, or SIPA. I will explain the potential role of stem cell myocyte in SIPA, and I will also show how these cells can be potentially used in the assessment of individual patient risk. So what is drug-induced arrhythmia? In a healthy patient, QT interval on electrocardiogram can be prolonged after administration of some drugs. QT prolongation is not harmful by itself, but it can be a precursor to a rare but very serious and potentially life-threatening cardiac arrhythmia, torsade de pointes, or TDP, or torsade. On a cellular level, cardiac electrical activity is described by an action potential. When an ventricular cardiomyocyte receives an electrical stimuli propagating from the neighboring cells, this cell depolarizes from its polarized negative membrane potential, that there is a plateau phase, and then cell returns to the resting state. This plateau phase corresponds to the QT interval on AKG that is supported by balanced inward sodium and calcium ion currents and outward potassium current. Most QT prolonging drugs act by binding hurt potassium channel that leading to the prolongation of action potential, or QT interval, And that can further lead to the generation of early after-depolarizations or arrhythmias that can be detected in induced pluripotent stem cell cardiomyocytes. So it was long recognized that some cardiac drugs cause torsade, but in the 1990s there was an epidemic of drug-induced torsade from non-cardiac drugs, such as cisapride, which is prescribed for acid reflux, or terfenadine, which is an antihistamine. Between 1988 and 2009, 14 drugs have been recalled from the market worldwide due to QT prolongation and torsade risk. There was an urgent need to develop a program to prevent drugs with unrecognized propensity to induce torsade from coming to the market. So International Conference of Harmonization adopted 2 guidelines that Food and Drug Administration currently uses as a guidance on how to assess the risk of drug-induced torsade. ICH S7B is a non-clinical evaluation of QT interval prolongation by human drugs based on assessment of hERG, potassium block and action potential, and QT studies in suitable animal species. ICH E14 is a thorough QT study typically conducted in healthy volunteers and applied to almost all new drugs. A change of only 5 milliseconds in QT duration considered significant under this study, and 10 millisecond prolongation is a positive thorough QT study that would require an extensive cardiac monitoring in phase 3 studies and would affect drug labeling. So adoption of these 2 guidelines has resulted in no drugs with unrecognized torsade risk being approved or removed from the market. So it worked, but it likely occurred at a cost. And this is why. First, hERG block and QT prolongation are not perfect predictors of torsade risk. There are multiple examples of the drugs that block hERG but are not torsadogenic. For example, verapamil, that in addition to hERG potassium channel also blocks L-type calcium channel, has been on the market for decades with no association with arrhythmias. Similarly, ranolazine, which in addition to hERG block also has late sodium channel block, is not proarrhythmic. So in drug development, though, hERG signal often means that drug is dropped from further development or interferes of, drug will have QT effect, and that will result in significant drug development challenges and regulatory hurdles. That leads to the situation when potentially useful drugs never get evaluated in humans. So how can we save these hERG blocking but potentially safe and useful drugs? We now have a deep mechanistic understanding of the ionic factors that result in torsade, and we can support the new approach to assessing proarrhythmia risk beyond hERG block and QT prolongation. This new paradigm is called SIPA. It has 4 components. First, the drug effects on multiple cardiac ionic currents will be assessed using patch clamp experiments in overexpressed system using animal cells. Then that information would be used in a computer model of adult ventricular electrophysiology, and the net effect of all the channels would be predicted. The drug effects can further be tested in an integrated human stem cell-derived cardiomyocytes, and finally used human AKG phase 1 data to determine if there are any unexpected effects. So the goal here is to develop a new in vitro paradigm for cardiac safety evaluation of new drugs that provide more accurate and comprehensive mechanistic-based assessment of proarrhythmic potential. So I have been mostly involved with the third stem cell part of the SIPA. It was enabled by the discovery of stem cells and development of electrophysiological assays, allowing for the first time for quick and easy assessment of drug effects in human model in high throughput manner. These limitations to the stem cells myocyte model, including the immature phenotype and variability in the baseline characteristics and drug response of the cells from different manufacturers, also batch-to-batch variability from each cell provider. With all these limitations, the idea of direct assessment of drugs in human cardiac cells as opposed to animal models or human clinical trials is very attractive for the community. So at the FDA, we conducted a proof of concept study to assess the feasibility of such assays to help optimize the design and experimental protocols for the future SIPA validation study. We have studied 26 drugs in a blinded fashion with known torsade risk. We used 2 commercially available stem cell lines, iCell cardiomyocytes and Core4U cardiomyocytes. We used 2 plate-based platforms selected for SIPA myocyte study, microelectrode arrays, or MEA, and voltage-sensitive dyes, VSD. We studied both acute and chronic effects of the drugs, and we also looked at 4 ion current effects with manual patch clamp for potassium, sodium, lead, and peak sodium, and calcium currents. So here is an example of action potential recordings at the baseline, and after drug addition, you can see the widening or prolongation of action potential duration, and also drug-induced arrhythmias at high drug doses. So the graph on the right combines drug-induced effects on clinical QT, shown in blue, and stem cell effects, gray and orange. So the dose of the HER-blocking drug, the Fetolite, is on the x-axis, and the Fetolite-induced repolarization prolongation in milliseconds is on y-axis. We could obviously go much higher, to much higher doses in vitro assay than in clinic, and you see that at high dose, the Fetolite-induced arrhythmia is observed in stem cells, shown with stars here. So we did this type of study for all 26 drugs. 20 of those drugs in the study were associated with QT prolongation, and 14 with torsade risk based on FDA-approved label, and 6 drugs on the right in our study were chosen as negative control with no clinical TDP risk. So in our study, out of 20 drugs with QT prolongation, induced action potential prolongation, stem cells, cardiomyocytes, 17 when we used voltage-sensitive dyes platform, shown in bold, and 16 when we used microelectrode array platform. Out of 14 drugs associated with torsade risk, 10 drugs induced arrhythmias in stem cells, shown in red. Finally, no repolarization prolongation of arrhythmias were observed for the negative control drugs. The results of this study have been recently published in Toxicological Science Journal. Overall, the study demonstrated 100% specificity of the stem cell-based assay for drug-induced proarrhythmic potential, with sensitivity varying from 50 to 80% depending on cell type and platform used. So those were promising results, but coming from a single lab, they do not address the potential site-to-site variability when proarrhythmic potential of the drugs would be assessed at multiple experimental sites. So Multiside study was designed by the SIPA myocyte working group that I'm part of and was coordinated by Ksenia. There were 10 core sites and coming from US, Europe, and Japan, and 20 plus non-core sites. We looked at 5 different devices, either MEA or VSO, and we used 2 commercially available stem cells. So following their standardized protocols, these sites assessed 28 blinded drugs with known clinical records. They're shown here by the risk category that was defined by the panel of experts, cardiologists, including some drugs that had been recalled from US market or elsewhere, shown in bold here. The question that was addressed by this study were really how consistent the stem cell-based assays are and how variable would be results from the independent sites. So we don't have the results of this study published yet, even though we have all the data and analysis is being done. But this is a brief summary of the status. We did complete pilot study first with 8 drugs completed, and manuscript is ready for publication, I think. So phase 2 validation study with 28 drugs is also completed. Data is being analyzed, and results will be compared with prospective in silico reconstructions and categories of high, intermediate, and low risk of torsade, and role of myocytes in regulatory proarrhythmia risk assessment will be defined. So I wanted to spend the last few minutes talking about our ongoing FDA study exploring the role of stem cell cardiomyocyte in personalized medicine. So in SIPA, we're using commercially available iPS cardiomyocytes that were originally developed from 1 or 2 persons to assess the general population drug response. Drug response can actually vary from patient to patient. Now, since these iPS cardiomyocytes can be easily derived from any patient using simple blood or skin sample, they do carry donor-specific information. And the question is, what if we could use patient-derived cells to predict an individual clinical outcome? So in 2014, Dr. David Strauss' group at FDA conducted a clinical trial looking at the 20 healthy volunteers' response to 2 QT prolonging drugs, the dafetalide and moxifloxacin. On the top graph here, the dafetalide concentration is on the x-axis and drug-induced QT prolongation is on the y-axis. The average population response to the drug is shown in blue, and you can see that individual patients shown in red then can have much lower response, which we call low responder here. So on the right is a high responder shown in red again, and on the bottom you see representative EKG recording from both low and high responder from this clinical trial. So what if we could predict which patient will benefit and which likely to be harmed by a drug by testing first in the patient-specific iPS cells? So what we did, we collected blood from all 20 consented subjects in the clinical trial, and we used 2 outside laboratories to reprogram blood cells into stem cells and then to differentiate them into iPS cardiomyocytes. Then we tested the same 2 drugs that subjects received clinically on the individual iPS cells using 2 electrophysiological assays. And after that, we would like to investigate if there was a correlation between individual clinical response to dafetalide and moxifloxacin with in vitro response of these drugs in the healthy subjects. So we planning on publishing our findings from this study by the end of the year, but in the meantime, I have included 2 references to the prior papers confirming the feasibility of this approach. In the first, authors were able to separate patients into low and high responders groups based on the subject-specific iPS-derived cardiomyocyte responses to sotalol. In the second study, the predisposition of breast cancer patients to develop chemotherapy-related cardiac side effects correlated with patient-specific iPS cardiomyocyte response to doxorubicin. So there is a glimmer of hope for detecting patients that are likely to be harmed by a therapy before actually giving it to the patient, but instead testing it first in patient-derived stem cells. And again, these cells only require a simple blood draw for the cells to be generated. So in summary, I was talking about how FDA regulatory research supports review of emergent innovative products, including induced pluripotent stem cells, how we develop alternative approaches to safety testing. I've talked about my side part of SIPA initiative and how that may soon affect the way cardiac safety of all new drugs is assessed by the FDA. I touched upon the potential uses for stem cell in personalized medicine and predicting dog clinical outcomes. I hope the case study that I have presented gives you an example of what questions can be addressed with regulatory research at the FDA and illustrate what a tremendous effect on medical product development and approval these studies could have. I will end by acknowledging my FDA colleagues and industry and academia collaborators, stem cell providers, SIPA contributors. We are always open to developing new collaborations. Please feel free to contact me if you think you may have research questions that might be of equal interest to the FDA. Now, thank you very much for your attention, and Dr. Ribeiro will be happy to take any questions you may have.
Thank you both for your opening remarks. Starting with this talk and for the rest of the program, we're going to have a format that will start with a presentation, and then after that, enough time for Q&A. So this will be the first session we do that with. I think we found that a lot of the value you get out of the workshop are actually the questions you ask and the questions that are asked by your colleagues. So don't be shy about that. Just to explain the setup, in the room, I'll start first. We have 3 mics positioned in the hallways, in the aisles right here. And feel free to come up now if you have questions about this talk or in the future sessions. Feel free to do that at the end of the talks. And for those of you who are following online, you can be entering your questions at any time. You don't need to wait to the end of a session to do that. You can be typing it as you hear prompts, thought during remarks said during the session. So that's sort of the format we'll follow for this and the other talks as well. So to get us started, I will turn it over to one of our DICE colleagues, Diane Nell. Looks like we may have some online questions. So Diane, can you read off the first question you have for us?
How does the FDA choose what research projects to work on, and may I make suggestions?
Maybe Alexandre, you start off first.
Yeah, I can start, at least in my division. We are normally guided by what's new in the scientific field. Most of what we do is published. Only a small percentage of the work that we do is confidential because it's related to reviews. We, of course, do not research anything. It's not a basic science division. We do applied science. And it needs to have an application for evaluation drugs, efficacy, safety, preclinical, clinical, etc. How can anyone contribute? We're completely open. Our emails are publicly available. We are open to any contribution from anyone in an academic or industrial or public environment. And you know, as Ksenia mentioned, feel free to email me, and I've been in contact, and other people in my division have been in contact with academics and research and also industry. What happens after we initially engage, we establish agreements, collaboration agreements, or memorandum of understanding, and then anyone who wants to collaborate with us can collaborate via those agreements. You know, it has to have we must develop a research plan that is clear and really well specified, and that is beneficial for the mission of the FDA.
Maybe I'll add that often the new research projects they stem from an application that came to the reviewer division or reviewer office. So how the science office interacts with reviewer offices, we do consults for them. So if there is a new emerging technology that maybe nobody dealt with before in the reviewer division, they go to the science division, and then there are usually scientists who are experts in specific area, and this will actually happen with this product in my research division, because there was application trying to use induced pluripotent stem cells as in vitro diagnostic device that we started to look at very carefully what this technology is, and that led to more and more sophisticated experiments we've done. So this is another way how new research project can be initiated in OSAL or other centers.
Thank you. Diane, can we have our maybe next question?
This is a question for Dr. Blinova. For the studies you presented on iPSC-derived cardiomyocytes, how did you address the issue of cell maturity? Did maturity of the iPSC-derived cardiomyocytes affect your results?
This is a great question. Really, the main disadvantage or drawback of these cells, existing cells, is the maturity. They really resemble more fetal cardiomyocytes rather than adult cardiomyocytes. But as our study shows, those are still human cardiac cells, and they express most of the appropriate ion cardiac channels. And as a fit-for-purpose model, they can be really good. There are some limitations for some drugs that affect specific channels that are underexpressed in this iPS cardiomyocyte. The model may not be very predictive. But in general, this is a great model. And also, there's a hope. This is a very new field. The induced pluripotent stem cells are only, a decade old, yes. Right? So this is a very rapidly developing field. And I know that cells changed dramatically from 5 years ago. And now they look different. They have different morphology. They look more mature. And Alex works on, specific devices that can also mature those cells and make them closer to real patients' heart cells. So we are very optimistic in this regard.
Thank you. Looks like we have a question from the audience.
Hi, I enjoyed both of your.
I think it's on.
I enjoyed both of your presentations, but I was curious on the personalized medicine, the clinical trial that Dr. Strauss led. How long does it take from the blood draw to having the actual results to be able to know whether or not you can apply the therapy?
Can you read the question?
Yeah, sure. So the question was, if to use iPS cells for diagnostic, how long does it take from drawing blood to actually have cells ready for any drug assessment or anything like that? It does take time. It's not a rapid test. As Alex slideshow, I think it takes at least 30 days to differentiate cells, and it's additional few weeks at least to do stem cells out of blood. So it's not a rapid and immediate thing to do. But in the future, you can make those stem cells, freeze them until you need them, and they remain, specific to the donor for the rest of his life. So this is one of the opportunities. It's not a rapid test. It takes
the quality.
time.
The quality of the induced pluripotent stem cells is a key factor. So when we reprogram somatic cells from blood or from skin, and we originate induced pluripotent stem cells, we must ensure that, for example, we must karyotype the cells. We must genetically characterize the cells. We must also ensure that the key list of quality factors that are now defined that basically define a good quality for a line of pluripotent stem cells. So that also takes a lot of time. A big issue right now is to do genetic deep sequencing. Mainly in my field, where we want to look at general safety issues, we must make sure that the lines that we are using, they don't have genetic defects that cause specific types of disease or a specific type of reaction to drugs. So for that, we also need to do deep genetic sequencing, and that takes also a lot of time. It basically depends on what you want to do and how deep you want to go on the information that you want to obtain from these assays.
That's probably won't be the mainstream assay, for everybody, all healthy patients. But if you have, maybe a child with a genetic disease or, patient really at risk for cardiac effects, it may worth it. Waiting a couple of months before starting a new therapy can be very harmful in finding, on the dish first, the drug that will be safe for the specific patient.
Thank you. We'll take one more question, I think, online. But before we do that, I did want to show this, point out on the slide that's shown here. You will see a survey link for each of the presentations that are delivered. There's also one for the overall program that we'll ask you to complete. And as Brenda said earlier, we really encourage you to share your feedback for us on each of the talks and topics you're interested in for us to deliver in the future. So I just wanted to point that out there. So with that, let's perhaps, Diane, take our last question for the session.
I'd like to do some research with the FDA, either as a fellowship or just as a one-time collaborative project. Is there a process for doing this?
It really depends on the there are ads for jobs at the FDA. There are programs for postdocs, similar to what the postdoc is, which is an ORISE fellow, ORISE programs. These are you can find them, you can Google it and find ads for different positions at the FDA, and I'm pretty sure there are many now that are available, and there are always a lot of them that are available.
You're hiring, right?
Yes. At the same time, it completely depends on the level of the candidate. If someone with a PhD has higher probability of doing research at the FDA, it completely depends on the capabilities. But there are many opportunities for anyone to work at the FDA. And in terms of volunteering, let me know and I can talk to HR. I'm new at the FDA. I haven't been here for a year, so I don't know all the details. But I can talk to HR and figure out how we can work with volunteering. It's something that, I don't see why that wouldn't be possible, but I don't know the details.
Other collaborative opportunities where one may reside in their academic or clinical research settings and then partner without needing to.
Absolutely. There are specific, programs that we can collaborate with academia and companies. We have several research collaboration agreements in place right now with different players. So that's certainly a possibility. It needs to be on the paper, how we collaborate and what comes out of the collaborative research and who owns the data and who publishes the data. But it's definitely possible, and we're very interested in this.
All right. Thank you so much. That'll conclude this session. Thank you, Alexandre.